Hello guys, this is my new channel and today I am going to discuss about mycoplasma. The mycoplasma is the smallest free living microorganism that can be grown in cell-free medium. So this is the structure of mycoplasma which lacks cell wall. It is a three-layer cellular membrane, that is trilamellar membrane. This part is called as the cytoplasm, and these teeny structures are called as soluble RNA. This granular structure is called as ribosome, and this is the double helical structure of DNA. This mycoplasma also contains soluble protein. So let's come to the definition of mycoplasma. Mycoplasma are a group of bacteria that are devoid of cell walls, so they are highly pleomorphic. Pleomorphic means they can change their shape and can pass through different filters, and they have no fixed shape and size. They lack the cell wall precursors like muramic acid and diaminopimelic acid, and the cells are bounded by soft trilamellar unit membrane containing sterols. Here you can see the trilamellar structure of mycoplasma, and this also contains sterols, and they have plasticity, so they can pass through bacterial filters and often have been mistaken for viruses. As they can pass through various filters, previously people think that they are viruses. And about the history of mycoplasma, the first member of the group was identified was the organism causing bovine pleuropneumonia, which was isolated by Knockard and Rocks in 1898. Many similar isolates were also obtained from animals, human beings, plants, environmental sources. They came from, and they are called as pleuropneumonia-like organism. But the new term of that organism is mycoplasma. Now people call this as mycoplasma. Then comes about the morphology of mycoplasma. Mycoplasma lacks cell wall and can pass through filters of 450 nanometer pore size. They occur as granules and filamentous of various size and structures. A distinctive feature seen in some species is a bulbous enlargement with a differentiated tip structure by means of which organisms attach themselves to suitable host cells carrying neuraminic acid receptors. This neuraminic receptor is found in mycoplasma and it helps in attaching to other organism or other structures. And mycoplasma do not possess flagella, fimbriae or spores, and they exhibit gliding motility. Gliding motility means they can attach to other organisms through this type of motility, and they can pass through various other places or other membranes, etc. Mycoplasma are gram negative and the stain by GMs are stain. And about the culture, mycoplasma can be cultivated in both liquid and solid media. They are generally facultative anaerobes. You know about anaerobes, which don't need oxygen to grow. And these are facultative anaerobes, means they can live in the presence of oxygen also, and their growth occurs better anaerobically. They can live in oxygen, but the better growth occurs in without oxygen environment. They grow within a temperature range of 22 degrees Celsius to 41 degrees Celsius, and the parasitic species growing optimally at 35 to 37 degrees Celsius, which is our normal body temperature, and saprophytes of mycoplasma grow at lower temperatures. Media for cultivating mycoplasma are enriched with 20% horse or human serum yeast extract, and sometimes penicillin and thallium acetate are added as selective agents. Higher concentration of serum is necessary as high source of cholesterol and other lipids, and you can see the appearance of colony after the incubation for 2 to 6 days and are about 10 to 600 micrometer in size. and their colony is typically biphasic with a fried egg appearance consisting of central opaque granular area. This you can see the opaque granular area, and the growth extending into depth of the medium surrounded by a flat translucent peripheral zone. This is showing the peripheral zone of Mycoplasma, and this is the opaque layer, and dense is the granular layer. This is the granular layer. So this figure is showing about the fried egg appearance of Mycoplasma. We can also see this appearance if we culture Mycoplasma under laboratory conditions. And various biochemical reactions required for Mycoplasma are their chemoorganotropes, and their metabolism is fermentive. Most species utilize glucose and arginine as the major source of their energy, and this urea is not hydrolyzed by them except by ureaplasmas. They are generally proteolytic, and Mycoplasma lack the ability to synthesize purines and pyrimidines. And about which their resistance, about up to which temperature or like that, Mycoplasma generally resemble non-sporing bacteria in heat resistance, but some strains are more sensitive and being destroyed in 45 degrees Celsius in 15 minutes. In only 15 minutes you can destroy them by rising up temperature up to 45 degrees Celsius. They are relatively resistant to lysis by osmotic shock but are very sensitive to lysis by surface active agents and lipolytic agents such as taurocolate and digitonin. They are resistant to penicillin and cephalosporin as well as lysozyme that act on bacterial cell walls but are sensitive to tetracycline and many other antibiotics. These are various antibiotics, means they are very resistant to antibiotics. And antigenic properties, which antigenic properties they have? Serological tests, these are the tests which we make to ensure that one person have infected with Mycoplasma or not. Serological tests such as complement fixation, agglutination, passive hemagglutination, ELISA, and immunofluorescence have been used to detect antibodies in sera and to identify isolates. About the reproduction procedure, multiplication is done by binary fission, but as genomic replication and cell division are often asynchronous, budding forms and chain bits are also produced. It means there are various, more than one feature for reproduction. And about the classification of Mycoplasma, they have been placed in class molecules, order Mycoplasmatis, which contains the following. They belongs to family Mycoplasmataceae, family Acoliplasmataceae, family Spiroplasmataceae, family Anaeroplasmataceae. And about the pathogenicity of Mycoplasma, they generally produce surface infection by adhering to the mucosa of respiratory, gastrointestinal, and genitourinary tracts. Generally, they found in gut and our respiratory system, mainly in mucosa of respiratory system. Mycoplasma can cause two types of disease in humans, that is pneumonia and genital infections. Laboratory diagnosis used to detect Mycoplasma or the growth of Mycoplasma can be easily screened by tetrazolium reduction test, in which Mycoplasma colonies reduce the colorless tetrazolium to red color formazan. And cold agglutination test, the agglutination test is also done for Mycoplasma. And this is all about the topic Mycoplasma, and I can make more topics on, if you want any more topics then you can comment me, or I have my own Telegram channel, you can message me there also. I will make other videos for your understanding, better understanding. And this PDF you can get in Telegram channel, in this name, Lifesciences with SS. I have a new Telegram channel, you can join there and message me for this note. It is in form of note, you can write in exam also, this is a good note, so you can comment me. And if this video is useful to you, then you can like, share and subscribe. Thank you.